Okay, as mentioned in class, the following lecture is part of a flipped classroom experience in which you will listen to the lecture first and then obviously take notes, and then we will have a quiz, a question and answer session in class, followed by some problem solving. So please do pay attention to the material presented in this lecture. So today we're going to cover gases, gas exchange, and acid physiology of the blood. Pretty much all of this comes under the umbrella of respiration. So we will focus on the respiratory mechanisms and structures that animals have developed throughout evolution. Okay, so this class is going to start with some considerations about evolution of the respiratory system in humans or higher vertebrates. We will discuss the property of gases as they pertain to respiration. Then we will cover several different examples of gas exchange membranes that we can find in different phyla of animals. And finally, we will talk about the transport of gases, both oxygen and carbon dioxide in the blood, and how this all pertains to the regulation of acidity of the blood. All right, so let's get started with evolution. Now, as I did before in the lectures, I'm basing myself on a single publication, and again, this is not meant to be an exhaustive review of what we know about the evolution of gas exchange membranes in animals. This is merely a starting point to think about gas exchange membranes and how they may have come about. So the first few ideas are based on this review called Structure, Function, and Evolution of the Gas Exchangers, Comparative Perspectives. Now, in this review, the author talks about several necessary steps that had to occur for the evolution of gas exchange membranes in animals. And the first of those steps occurred a long time ago, and it had to do with a switch of metabolism from anaerobic to aerobic metabolism. That was the first step, and it was brought about by this huge revolution caused by cyanobacteria that started to enrich the atmosphere with oxygen. All right, now the second step was the evolution of multicellularity. Now, a unicellular organism is directly exposed to its environment, so it doesn't really need a gas exchange membrane. Well, basically, the cell membrane is the gas exchange membrane. Now, talking about vertebrate evolution, the next step would be the evolution of a closed circulatory system. A closed circulatory system that, as we have seen, is very efficient in supplying different tissues with oxygen and to adjust the delivery of oxygen to the tissues and nutrients as well. The fourth step would be the origin of a carrier pigment, a respiratory pigment such as obviously hemoglobin, but there are others as well. Then, as we continue the evolution, as we have seen before, if we need to increase surface area, And in the case of respiratory surfaces, we are interested in increasing surface area because a greater surface area means more gas exchange. So the next step is the formation of invaginated respiratory organs. Now, this particular step made it possible for animals to make the move from water onto land. Now, the next step is this movement from water to land, in which now lungs become the principal respiratory organs rather than gills. Then in step 7 we see the development of a double circulation. Now remember that the double circulation creates not just a systemic circulation that takes blood to most of the body, but it also creates a pulmonary circulation. And this pulmonary circulation is the one that picks up oxygenated blood to transport it to the rest of the body. And having a separate circulation for the pulmonary system and for the systemic circulation means that now we can actually pump blood into the pulmonary circulation at a lower pressure, and thereby pick up more oxygen and prevent all these vascularized tissues around the lungs to suffer any damage. Then we will see, and this is actually something that we're gonna see in this lecture today, is in terrestrial animals, first respiration occurs through buccal force pumping, in which a positive pressure is created that forces air into the lungs. Now later on we see that the evolution of a suctional breathing as in human beings, where air enters the lungs passively due to changes in pressure around the lung. Now we're going to explain this later on. Then in step number 9, we will see a progression from ectothermic heterothermy. So those are the animals that oftentimes in common parlance are referred to as cold-blooded, but I already told you that this term is not really appropriate. Now we move from ectothermic heterothermic animals to endothermic homeothermic animals, and that means a huge step up in metabolic requirements and more metabolism that is aerobic based on oxygen means that we need more efficient respiratory surfaces. And then finally, in some vertebrates, as in birds, we will see that the respiratory system has attained great efficiency. Now, birds, as you may have heard occasionally from me, have a very high metabolism. And a very high metabolism means that we need a great capacity to exchange gases. So we will see a little later on how that particular respiratory system in birds actually looks like. Now, all in all, though, we see this progression, this evolutionary progression of gas exchange membranes. All respiratory organs share some common features. First, we have a moist surface in which gases can dissolve and diffuse. Now, this is a no-brainer for aquatic animals. Of course, they have a moist surface. I mean, they're in the water. But this is also true for terrestrial animals. All terrestrial animals have a moist surface, as we will see later when we discuss the mammalian lung. 2, we have an increased surface area for gas exchange. All this invagination, all this folding, the formation of very fine and large surfaces where gas exchange can take place. Third, we have a great amount of blood flowing through gas exchange membranes. The more blood flows through gas exchange membranes, the more oxygen and carbon dioxide can be transported across the respiratory surface. And finally, all respiratory organs have a very thin and delicate structure. And the thinness is paramount to respiration. Why? Well, I'm not going to tell you right now. I just want you to think about it for a little bit. Why is it so necessary that respiratory organs have a thin and delicate structure? So here we can see a hypothetical respiratory surface with respiratory organ cells up here, and I'm just going to activate a pointer that makes it easier to see. Alright, so these are respiratory organ cells that are generally fairly thin. Then on the inside we have our blood vessels. These are capillaries with a simple endothelium and with a very small diameter. A diameter that is almost barely enough to allow for the passage of red blood cells. And then obviously here on this side we have our hypothetical tissues that require the exchange of carbon dioxide and oxygen. Okay, so the next step is to talk about the property of gases in as much as they pertain to respiration. So this will not be, oh, hang on the clock. Okay, we're done. So this will not be a chemistry class on gases. This is simply a very basic lecture of properties of gases that are important for respiration. Now, first of all, what does air that we breathe in consist of? Well, it consists of about 78% of nitrogen. This is elemental nitrogen that is biologically speaking pretty inert. Then we have 21% of oxygen, which is more like 20.93% of oxygen. Then we have some other gases that are present in lower quantities, and one of these gases, even though it makes up only 0.03 to 0.04% of the atmosphere, is of utmost importance for life on the planet, and obviously important for respiration, and that is carbon dioxide. Now, usually when we talk about gases in the atmosphere, how do we express quantity? Well, we don't really talk about concentrations, but we talk about pressure. The amount of pressure that these gases exert in the atmosphere. Now, atmospheric pressure is usually measured nowadays with a standardization of units as kilopascals. But traditionally, especially in the United States, it was expressed as millimeters mercury. So Hg is the elemental symbol for mercury. Now, just keep in mind that 1 kilopascal is roughly 7.5 millimeters mercury. Now, at sea level, so this would not be the case in Yachaitek at 2,100 meters elevation. At sea level, the pressure is about 760 millimeters mercury, or about 100 kilopascals. Now, this is at sea level. The atmospheric pressure will decrease as we move up the mountains. And we can see a profile of how the pressure, the atmospheric pressure, changes with elevation on this particular graph. So down here on the x-axis we have meters above sea level, 0 being sea level, and then on the y-axis we have atmospheric pressure in millimeters mercury. And we can see that there is a steady decline in atmospheric pressure with elevation. Now, right here, well, let's move back up here. This is roughly where Yachaitek lies in the in the highlands of Ecuador, so about 2,100 meters above sea level. And here the atmospheric pressure is slightly above 600 millimeters mercury, so it has decreased almost by 150 millimeters mercury. Then, as we go up higher the mountains, we will see a continued drop in atmospheric pressure. Now, right here, right about here at 4,600 meters, that is equivalent to the altitude or the elevation of Taita Imbabura, the volcano that's sitting right in front of us. Here, the atmospheric pressure is down to about 440 millimeters mercury, or we could also say it's a decrease of 320 millimeters mercury in terms of pressure. Down here, further down, the highest mountain of Ecuador is somewhere around here, and here we have less than half the atmospheric pressure that we had at sea level. And obviously, we continue to drop and continue to drop the higher we get. So once we get up to Mount Everest, which is at 8,848 meters, the amount of oxygen, or the pressure of the atmospheric pressure, and therefore the amount of oxygen in the air, is right at the limit of what a human being can experience. Now, if Mount Everest were 100 meters higher, then probably humans could not get up the mountain without any oxygen masks. Maybe I'm exaggerating, maybe it's 200 meters, but that's not the point. The point is it's right at the limit. Now, the total amount of atmospheric pressure is actually the sum of the pressure that each individual gas in the atmosphere exerts. Now, if the total amount of atmospheric pressure is 760 at sea level, and we have an oxygen content of roughly 21%. That means that the contribution to the total pressure in the atmosphere by oxygen is about 160 millimeters mercury. So this is the amount of pressure that oxygen exerts in the atmosphere. Now, for the partial pressure of nitrogen, Well, that would be about 0.78 times 760 millimeters mercury. Now, what's interesting is that as we go up the mountains, even though the atmospheric pressure continuously drops, the contribution of each individual gas to the atmospheric pressure remains constant. So regardless of altitude, the percentage of oxygen is always going to be around 21%. It's going to be 21% at sea level. It's going to be 21% on Mount Everest in the Himalayas. So it never changes. But obviously in the Himalayas we have a very low total atmospheric pressure. So there is going to be much less oxygen present. Now, another thing that is important to keep in mind is that the transfer of oxygen as well as of carbon dioxide across the gas exchange membrane is driven by partial pressure gradients. Oxygen will move down its pressure gradient from high pressure to low pressure, and carbon dioxide will do exactly the same thing. Now that means that if in the air we have or in the atmosphere we have a lower pressure of or partial oxygen pressure, at high elevations diffusion, the rate of diffusion will be much slower. So not only is there less oxygen around, but diffusion will be slower. Okay, now let's get to another important characteristic of gases as they pertain to respiration: the solubility of gases. And this is something that we've already touched on in previous classes. Now, all gases that are in the atmosphere and that come in contact with a solution, such as fresh water, seawater, or body fluids, all gases will dissolve in a watery medium. There's some diffusion always going on. Now, most gases dissolve poorly in water, but as we are going to see, our body has mechanisms to keep the pressure gradient fairly large and thereby continuously encourage diffusion across the gas exchange membrane. Now, there are several factors that we need to consider that are important in influencing the solubility of gases in water. The first one is atmospheric pressure. Higher pressures will result in more gas in solution up to a certain limit for each gas. So, if we have a glass of water sitting out on the table, maybe on the beach overlooking the ocean, the atmospheric pressure is going to be fairly high, and the amount of oxygen in that glass of water is going to be also fairly high. If we are sitting on top of Mount Everest with our little glass of water, just assuming for a moment that the water will not freeze, because of the amount of oxygen in the air, there will be much less dissolved in this water. So higher pressure means more gas. Now, the next point may be unknown to you, or you may have forgotten about it and probably learned about it in chemistry and ever since have forgotten about it, is that cold water holds more gases than warm water. Now, I mentioned that in the last class, and that means that more gases will dissolve into water when the water temperature is low. Cold water will have the ability to hold more oxygen, more carbon dioxide than warm water. Now, another factor that determines the solubility of gases in water is the amount of other solutes that are present in solution. And these will variably affect the solubility of gases. Usually, the more solvents there are, the lower the solubility is. So clean water can hold, without any solutes in it, can hold more oxygen or more gases than water with a lot of solvents. Water with a lot of solutes, not solvents, solutes. All right. So we talked about evolution, we talked about the property of gases. Now we will begin a long section on the types of gas exchange membranes in different types of animals. Here we go. Now, there are a few concepts or ideas that we need to cover before we actually go into the different types of respiratory organs. Now, first thing is, there are relatively few mechanisms for gas exchange. They all involve thin structures with a great amount of blood vessels, usually large surface area, and a moist surface, as we've seen before. And there aren't that many designs out there in nature that would really be useful. Okay, now on one hand we can have diffusion of gases across the body's surface. And we've kind of talked about that before. Think about flatworms in the phylum Platyhelminthes. Those just take up oxygen across the skin. They are flat, and flat is good because diffusion rates will be much greater over very short distances than great distances, as I explained previously. Now, a step up from that is the presence of specialized organs like gills, which we see not only in fish, but in several types of invertebrates, and invertebrates, fish. I'm trying to think, there are even some salamanders that have gills. Tracheae, which we are going to find in insects chiefly, and lungs, which is another important respiratory organ. Now, lungs we have also in mostly terrestrial organisms. Okay, now a term that we need to know, that we need to remember, is the term ventilation. When we talk about ventilation in respiration, we are talking about the process of bringing oxygenated water or air in contact with the gas exchange surface. So ventilation is equal to bringing oxygenated air or water across respiratory or gas exchange surfaces. Now, also before we get into the details of some of these respiratory organs, there are going to be very different selective pressures, or very different pressures in general, on gas exchange membranes when dealing with terrestrial versus aquatic animals. There are really different challenges to gas exchange in these two groups of animals. Now, there are a few considerations here. Aquatic animals have less available oxygen. That is because the solubility of oxygen in water is low. I've talked about this previously. So the total amount of oxygen in the water is going to be lower than the atmosphere. Another problem that aquatic animals face is with temperature changes. Remember that the solubility of oxygen and other gases depends on temperature. That means for an aquatic organism that lives perhaps, say, at 10 degrees Celsius, if for whatever reason the water should increase in temperature to about 20 degrees Celsius, the water is going to lose a lot of oxygen. And the fish that requires the amount of oxygen present in the water at 10 degrees Celsius now may be in serious trouble. So that is a very important consideration for aquatic organisms. Now, as for terrestrial animals, the main issue, the main problem is maintaining your respiratory surfaces moist. We need that moist layer, or this layer of moisture on our respiratory surface so that we can dissolve the gases into a liquid that then allows for the diffusion of these gases from this moist layer into our bloodstream. So the biggest challenge for terrestrial organisms is desiccation. And this explains perhaps as well why lungs are so efficient, and also trachea in insects are so, well, maybe efficient is not the right word, are so well suited to this desiccating environment. It's because our lungs are an enclosed space, and it turns out that the humidity, the relative humidity of the air in our lungs is completely saturated at 100% humidity, and that is what keeps our respiratory surfaces humid. Now, terrestrial organisms have also another great advantage. They have more oxygen available to them. Oxygen in the air does not fluctuate according to temperature as oxygen content in the water. It also turns out that ventilation is a less energetically demanding event in terrestrial organisms. To move air over our respiratory system or respiratory surfaces is so much easier than for aquatic organisms to move water over the respiratory surfaces. So for terrestrial organisms, breathing in air is energetically more efficient than breathing in water for aquatic organisms. Okay, so let me see. This last comment. Water is denser than air. Yeah, mentioned that. Removes heat from gill surface. That is also important. For fish, continuously the water that streams over the gills takes heat out of the body, if you will. And the water that moves over the gills is also important, not just for the exchange of gas, but the exchange of water and salts. There's a distinct osmotic environment or around the gills that is different from the osmotic environment inside of the cells, and that will cause water and salts to move as well. And that's something that we will get to when we talk about the excretory system. All right, so as I said before, some invertebrates, as flatworms, that are only a few cell layers thick, diffusion can take care of gas exchange. There's no need for a specialized transport mechanism. There's no need for a specialized respiratory surface. It also turns out that even though other means of respiration are important for larger animals, some, especially amphibians, do exchange a considerable amount of oxygen and carbon dioxide over their skin surface. Now we will get to that a little later. Well, actually, we get to it right now. Here we are. Here we have a group of amphibians, starting with lungless salamanders, bullfrog, mud puppy, which is a type of salamander or newt, turtle, eel. Okay, now turtle is obviously not an amphibian anymore. That's a reptile. The eel is a fish. Boa constrictor, which is another reptile. Green lizard, goldfish, another fish. The big brown bat. A trout, which is another fish. And finally, humans. And what we can see is where are we? Here we have our amphibians. They do exchange a very large amount of gases through their skin, especially lungless salamanders. That makes sense. They are lungless. That means there is not much more surface for them to exchange gases than their own skin, and the total amount exchanged is virtually almost 100% across the skin. Now in the bullfrog we see that yes, carbon dioxide is exchanged a lot. Oxygen is to a much lesser degree. And this means that these bullfrogs, they require lungs to a large degree to assure a steady supply of oxygen to body tissues. The mud puppy, well, it still exchanges carbon dioxide and oxygen almost at the same level through the skin. About 30 to 40% is still exchanged. Now, interestingly, in the turtle, which is a reptile, that's almost the same amount. So even a turtle may exchange a lot of gases, especially carbon dioxide, across the skin. If you look at the European eel, well, it also exchanges gases, but almost entirely oxygen. There's no carbon dioxide here. The boa constrictor, which is a reptile with dry scales, we would think, well, they don't exchange any gases anymore. Well, about 20% of their carbon dioxide is still exchanged over their skin. Green lizard, that's more. Goldfish, even less, and only oxygen. Bats, well, bats are still exchanging a little over 10% of carbon dioxide over their skin. Brown trout, a little bit of oxygen. And then, well, what is this? In humans, humans do exchange a little bit of gases across their skin as well. Less than 5%. We take up a little bit of oxygen throughout skin, and we get rid of a little bit of carbon dioxide as well. All right, so skin is a important respiratory surface in many different animal groups. Now, an additional respiratory surface due to extensive projections and foldings that create a large surface area, are external gills. So external gills are not carried inside of the body, not like a fish that has the gills tucked in safely on the inside. External gills are on the outside. They're exposed out into the medium. Now, these external gills, depending on the animals, they can be found in one particular area of the body, or they can be spread throughout the entire area of the body. Now, these surfaces are or do have some limitations, some problems. A, they're unprotected and subject to damage. Now, we've mentioned before that gills have a large amount of blood vessels. Now, large amount of blood vessels, if these external gills become damaged, that can potentially mean a very significant loss of blood. So very unprotected and subject to damage. I can imagine many animals out there, predators, looking for prey, and they see an external gill and they think, well, let's nibble on that first. The other downside of external gills is there is some energy required to move the gills in the watery medium. And they need to be moved. Why? Because if the gills remain in the very same spot in the water, they're going to exchange gases. But if the water around the gills does not move, then the water around the gills will become depleted in oxygen and enriched in carbon dioxide. There's just going to be a layer of water forming around the gills. So you need to move the gills out of this pocket of low oxygen, high carbon dioxide water into some water that's rich in oxygen and poor in carbon dioxide. You need to keep these gills moving. And that requires some energy, but it also may attract predators. I mentioned many times throughout this course already that predators use as a cue, a visual cue, movement. Cryptic species that are very well camouflaged will not be seen by a predator unless these cryptic species move. Now, a constant movement of gills then potentially may alert the predator that there is some yummy morsel out there in the water. Now, here we have two examples, one invertebrate and the other one vertebrate. So on the left-hand side, we have a nudibranch with a clustered gill tuft. Nudibranch are mollusks, oftentimes very brightly colored. And you know already what this bright color means, right? Buzzword, aposematic coloration, warning coloration. Yeah, sure, I'm waving my gills, and sure, there's a lot of blood in there, and sure, I could be yummy, but I'm also very poisonous, so don't eat me. So aposematism is very common in these nudibranchs that have external gills. On the right-hand side, we see a larval salamander. So this is a vertebrate that also has these outside, these external gills, almost on its throat and the back, that would be the neck, right? And we can see that these are feathery structures here as well, and these feathery subdivisions, they're obviously there to increase surface area. Okay, so we're moving along. Now we will talk about some internal gas exchange membranes, and we will focus mostly on vertebrates from this point on. We will talk about fish gills, We will talk about avian lungs, which I mentioned before, are extremely efficient in exchanging gases. And we will also talk about the mammalian lung.